Welcome to The Explainer. Today, we're diving deep. We're going to break down the incredible microscopic engine that powers your every single move. I mean, everything from a simple blink to an all-out sprint. So let's kick things off with a question. It sounds simple on the surface, but it's actually incredibly complex. How does a simple thought, like I'm going to kick that ball, actually turn into a powerful physical action? How does that intention in your brain become real-world movement? All right. To figure that out, we're going to follow the entire chain of command. First, we'll look at the engine of movement itself. Then, we'll meet the protein players, the real stars of the show. We'll trace the signal all the way from the brain, find the one essential key that unlocks everything, watch the molecular power stroke in action, and finally, see how it all resets to do it all over again. Okay, first up, the engine of movement. We're zooming way in here, past the muscle you can actually see, past the bundles of muscle fibers, all the way down to find the fundamental engine that makes everything tick. And here it is. This little thing is called the sarcomere. It's the basic functional unit of muscle contraction. Think of it as the smallest possible piece of a muscle that can still do the job. It's in these tiny repeating segments that all the magic really happens. Okay, I know this looks a little complicated, but don't worry. The key takeaway here is just how perfectly organized this whole setup is. This structure is actually what creates that striped pattern you see in muscle tissue under a microscope. The main things you want to pay attention to are the two types of bands. You've got the dark A bands, which hold the thick motor filaments, and then the light I bands, which have the thin track filaments. Everything else, just think of it as the scaffolding. The Z lines and the M line, they're just there to hold this incredible tiny engine together. And this is so cool because it perfectly shows us the main idea behind what's called the sliding filament theory. Now get this, when a muscle contracts, the filaments themselves don't actually get shorter. Nope. Instead, they just slide past each other. And as they slide, the whole sarcomere shortens up, those light I bands shrink, and that H zone right in the middle, it pretty much disappears. But look closely. The dark A band stays exactly the same length. That's the key. Okay, so we know these filaments are sliding past each other, but what are they made of? Well, it's time to meet the key actors in our little molecular story, the two main protein players that make this whole thing possible. First on our list is actin. This is the thin filament. And honestly, the easiest way to picture it is like a long, twisted rope, or maybe even better, a track. This is the track that our motor protein is going to move along. But here's the thing. Actin isn't just one simple protein. It's really a team of three working together. You've got the main F-actin chain, which is the track itself. Then, wrapped around it, you have this other protein called tropomyosin. Think of tropomyosin as a guard, and its job is to block the spots where the motor wants to connect. And the final piece of the puzzle? That's troponin. Troponin is like the lock that tells the guard what to do. And now for our motor. This is myosin, the thick filament. Its most important feature is this series of little heads that stick out. These heads are kind of like tiny oars on a boat, and they are just itching to grab onto that actin track and start pulling. So yeah, myosin is also a pretty complex protein, but the part we really care about for movement is the myosin head. This thing is the powerhouse. It's got two super important binding sites, one to grab the actin track and another one for ATP. And you know ATP, it's the universal energy currency for pretty much everything in the body. It's the gasoline that makes this entire molecular motor go. All right, so we've got our machinery all set up, right? We have the tracks, we have the motors, but how do we flip the on switch? Well, as you can probably guess, it all starts with a signal that comes all the way from your brain. And this happens incredibly fast, like a rapid-fire sequence of events. A signal zips down a motor neuron to the specialized connection point called the neuromuscular junction. Right there, the nerve releases a chemical messenger called acetylcholine. And that chemical message instantly kicks off an electrical wave, we call it an action potential, that just sweeps across the whole surface of the muscle cell. Now, that electrical wave has one primary, absolutely critical job, and that is to release the key. The one key that will unlock all of this amazing microscopic machinery. Okay, this right here is the magic moment. As that action potential spreads, it triggers the release of a flood of calcium ions. And that calcium, that's our key. It rushes in and binds to that troponin lock we talked about. This binding causes troponin to change its shape, and that change physically shoves the tropomyosin guard out of the way.
Finally revealing the active binding sites on the actin track. So the sites are unlocked, they're exposed, and now the myosin motor can finally get to work. This is it. This is where the actual force is generated. We call this the molecular power stroke. And this all happens in a really rapid, powerful cycle. First, the myosin head burns some ATP, using that energy to get into a cocked, high-energy position. Then it binds to the actin, forming what we call a cross bridge. Then, bam, the power stroke happens. The head pivots and pulls that actin filament inward. Finally, a brand new ATP molecule comes along and binds to the head, which makes it let go of the actin, and then the whole cycle starts all over again, ready to grab and pull again and again as long as that signal is active.
And just try to wrap your head around this. This isn't just happening one time. This tiny little rowing action is a perfectly coordinated storm of activity. It's happening millions of times, across thousands and thousands of sarcomeres, all in a fraction of a second, just to power one single muscle twitch. It's absolutely mind-blowing.
But a muscle that's always contracting isn't very useful, is it? So what happens when the job is done? The muscle has to be able to relax and get ready for the next command. Well, it's pretty straightforward, really. The entire process just reverses itself. The nerve signal stops, that calcium key gets actively pumped away, put back into storage for next time, and without calcium, that troponin-tropomyosin guard dog complex slides right back into place, relocking the binding sites. The cross-bridge cycle grinds to a halt. The filaments slide back to where they started, and just like that, the muscle is relaxed. So there you have it. From a single thought, down a nerve, triggering a chemical release, unlocking a protein, and firing up a molecular engine. That is the incredibly elegant dance of how a muscle works. It's what powers every single heartbeat, every breath, every step you take. Which, of course, leaves us with one last question to chew on. What happens when this perfectly tuned, microscopic dance goes wrong?